Let's have a closer look at DNA replication right now. I already made another video, but I think it was a little bit messy, so I'm going to give it a second try, and I hope it's going to be a little bit more organized in this time. So what I'm going to show is I'm going to show you the so-called the replication fork, and the replication fork, this is the actual place where the DNA is copied. And what you see over here in light blue, that is my original DNA. Of course, it is normally a double helix, so this part over here should be a double helix. I simplified it and I made two parallel lines. And of course, we've got the hydrogen bonds here in between. So if there's a, let's say, an A over here on one side, then we have a T on the other side. And if there's a G over here, then we have a C over here, and so on. You get the point, right? Okay, so that is, we've got the hydrogen bonds here. And what we're doing right now is the following. I've got over here different enzymes that are involved in DNA replication, and I'm going to tell you what they are doing and how the whole thing works. Okay, so first of all, this is our original DNA. Our double-stranded DNA is over here, and you see that over here on the right side, the DNA is separated. It's because the hydrogen bonds are separated because an enzyme called helicase is separating the two DNA strands. But before we go into the enzymes, we have to label the DNA, and what I'm going to do is I'm going to label this end of the original DNA my 3' end. There's a 3' OH group, and then this must be my 5' end. And remember that the 3' end has an OH group at the end. You don't have to know this right now, but it's just to clarify. And the 5', that's the phosphate part, right? Okay. And then if this is the 5', then over here we must have the 3' end, right? So we're going to label this here 3', and this is 5', and it's pretty important. Without this, you won't understand the way the DNA is replicated. So we have a phosphate over here as well, and an OH group over here. Yeah. So what we now need to do is the following. Let's go through these enzymes over here, and I'm going to explain you what they're doing. So helicase is the first enzyme that I'm going to talk about, and the helicase is sitting right here on the replication fork. Well, actually, it's much bigger, right? But I'm just keeping it simple. Okay, I'm coloring this in. If I make it too big, it's too difficult to see, so that's a little bit small, I have to admit. It's actually much bigger, but that's my helicase. Okay, helicase, and the helicase is moving, in this case, to the left. Okay, so I should maybe indicate that we're using an arrow that's moving to the left because it's moving along, and it's separating, it's unzipping the DNA. You have to imagine that the whole thing is like a zipper, okay? It's like the thing that's unzipping the DNA and breaking the hydrogen bonds, and then you're separating the two DNA strands. And now we've exposed the bases, so you can imagine if there's an A over here, there must be, of course, a T over here, but it's now separated, okay? There's a T over here, and we have another A over here, and a G over here, then there's a C over here, and A, and you get the idea, right? Okay. So it doesn't really matter for right now what bases we have, but just to clarify this. And surrounding, all around here, what we have is we have nucleotides. And I'm really going to simplify my nucleotide. I'm going to make them little lines here. So I have an A nucleotide over here, I have a T nucleotide over here, I have a G nucleotide, and a C nucleotide, and many more, of course. Yeah, another A, and they're just floating around here, right? Okay. Remember how a nucleotide looks like. It has a sugar, a phosphate, and the base. And I'm really simplifying it here, right? And of course, each nucleotide has a 5' and a 3' end. I'm not going to indicate this now, right? And now what we're going to do now is we want to copy the DNA, and we're going to start here at the very bottom, okay? And what happens now is the following. The helicase is separating, has unzipped the DNA, and now I'm jumping over here to DNA polymerase 3 over here, and the DNA polymerase 3 would now like to start copying the DNA over here. So it would like to read the T, and it would like to insert an A over here, but it cannot do that, okay? Because what I do not have over here on this new, I'm missing something over here. I'm missing a 3' end to attach a nucleotide to. You see, the DNA polymerase cannot simply start making new DNA here. It must already have a pre-existing DNA over here or something where you have an OH group, a 3' end, to attach the new nucleotide to, but we don't have that yet. Okay, so that's a little bit of a problem, right? No, it's actually not a problem, because what we have is we have a primase. This primase over here is an enzyme. It goes there and it starts to make an RNA primer. Okay, and to indicate that this is RNA, I'm making zigzag, and that's RNA. And RNA primer. And you probably already know what RNA is. It has a U instead of a T. It has ribose and not deoxyribose sugar. So it's a separate presentation on the differences between RNA and DNA. But to indicate that this is RNA, I'm drawing a zigzaggy line, and over here I have a 3' end now. So the primase is able to make RNA directly without having anything pre-existing there, right? And this is now like a starter, okay? Now the DNA polymerase 3 comes along, okay? And let's say I have a G over here, and it reads the G and says, wow, cool, I've got a G here, and now I can attach, I've got a 3' end over here, and now I can attach a C. And it will take the C from over there, and it will attach it over here, right? And then let's say I have an A over here, then it will take the T, and it will attach it over here, right? And it will continue doing this, and every time when it attaches a new nucleotide, I get a new 3' end. So we can continue to make the DNA, and this is I did not draw it right now, but over here you've got the the DNA polymerase 3, right? That's my DNA reading the original DNA and inserting the complementary base, and it's moving along like this, right? And then it's making new DNA, yeah. And we always have a new 3' end, and so that's not a problem. So it's continuing to make DNA, and it's kind of following along the helicase, yeah, and into this direction. That's the 3' end. So I've got an OH again, an OH group. Yeah? Yeah? So it can continue to make DNA over here by always reading the sequence. So I have a C over here. It has inserted a G. It has inserted an A C. Yeah, you get the idea. Yeah. Yeah. So I have over here, I have my original DNA, and this one is the new one. So it's semi-conservative replication because I'll keep the original and I have the new one over here, right? So that is and this strand, this is continuous replication, continuous, because you see as the DNA polymerase 3 over here just keeps on following along the helicase as it unzips the DNA, and you can also call this the leading strand. Strand. That's the easy one. Okay, that's the easy one because yeah, you would just keep on making DNA. The DNA polymerase reads the original, inserts the complementary base, and yeah, then we have a double helix again over here. But the problem is up here. What I'm going to do now is simply because otherwise it's getting too chaotic again. I'm just going to erase these lines here again. Okay. And what we now need to do is we also need to copy the top one, but here we have a problem, and the problem is that the DNA is anti-parallel, and that we can still only make new DNA from a 5' to 3' direction. Yeah, so that's all 5'. This is the 5' end. Yeah, and to 3'. Yeah, that's fine. And if we have to stick to this rule here. But this means that essentially we have to make the DNA the opposite direction here, because this is the 5', so that's the 3', and that's the 5'. We still have to make it like this, but the problem is you see this is going the wrong way, yeah, because we're opening up the DNA towards the left, but we can only make new DNA towards the right because we can only attach new nucleotides, yeah, here, yeah. That's how we can make it, yeah, and we always make a new 3' end. It's going the other direction here. But that's the wrong way because the helicase is opening it up here to the left. So the question is, what do we do? Okay, so that's what I'm going to explain now. Yeah, so I'm going to erase all of this again here. Yeah. So and what we have to do is we do the following. We start making the helicase unzips the DNA, and we again need our primase, and this primase will make over here now an RNA primer. And this RNA primer has a 3' end over here and a 5' end over here. So we have a 3' end. And what will happen now is that the DNA polymerase 3, the same one as we had over here, the same one, will go here now, and it will start making new DNA into the opposite direction. Okay, that's not a problem. So we have the 3' end over here, the 5' over here. So it's making it into the opposite direction, but we're not forgetting, we're unzipping it over to the left. But what happens is that every time when I have more space here, it will start making a new RNA primer, and it will start making it in shorter pieces into the opposite direction. Okay, so it will always make a new RNA primer here, and then it's going to make a short piece of DNA into the opposite direction. And as we open it up more, we add again a new primer and we make it into the opposite direction, right? So what we get is we get parts here that contain RNA the RNA primer, and then of course we have parts that have the DNA. And you know what, because of so much space here from the previous session, okay, I'm going to make the RNA over here, and that's the DNA. Yeah? And so what we have a problem here right now, because over here, this part here, this is exactly, that's RNA, right? That's RNA here. Yeah? And that's not supposed to be in there. So what happens is the RNA polymerase 3 starts making DNA, and until it hits the previous RNA primer. And so the DNA polymerase 3 hits and runs into the RNA over here, and this RNA shouldn't be there. So what has to take place is we have to remove the RNA and we have to replace it with DNA, and this is where DNA polymerase 1 comes to play. So the DNA polymerase 3, which was sitting over here, that's DNA pol 3, goes away. Okay, DNA polymerase 1 takes over, and the DNA polymerase 1 will remove now the RNA. It will cut the RNA. Oops! Now I also removed the DNA. It will. This is the DNA. This should stay. Okay, this should stay over here. Okay, that's hydrogen bonds, and it will now replace the DNA all the way until over here. Okay, so it will fill in the last base. So if you have a C over here and a G over here, and this is a T, then it fills in the A, right? So all the way up to the last base, the RNA is replaced with DNA. Okay. So in other words, the DNA polymerase 3 makes the DNA continuously, and and also over here, and also the DNA polymerase 1, it has also the ability to remove the RNA and to replace it with DNA. Okay, so we are almost finished now, but you probably see that over here we have a small gap. Okay, a small nick. So the last bond has not been filled in yet. Okay. And we need another enzyme to do this, and this is DNA ligase. And the DNA ligase will simply connect the DNA, the bond, and then we have a clean or continuous strand of DNA only without any RNA. Yeah? So that's the role of DNA ligase, is to seal the little gap over here, right? So we talked about almost all of the enzymes. The only one that I didn't talk about yet is this DNA gyrase. And and this DNA gyrase sits over here in front of the helicase. This gyrase is over here. Gyrase. And what this DNA gyrase does is the following: as the helicase, if it opens the DNA, the DNA is a double helix. So all the twists, all the windings are going to be pushed forward. What's going to happen is if we simply keep on opening the DNA, then the DNA is going to get entangled, and we call it supercoiled. Okay, if supercoiled means that the twists are going to move, the turns of the DNA are going to move further down as well as we open the DNA, and this causes the DNA to become twisted like this, right? It's supercoiled. And that's not good, and the DNA gyrase now relaxes the DNA again. It cuts the DNA open and it takes the turns out so that the DNA stays nice and loose to be opened. Yeah? So this is so basically the DNA, the gyrase over here in front of the helicase takes the tension out of the DNA, because as the helicase opens it up, the twists also start to move towards the left, and it's gonna supercoil the whole thing. Yeah. So that is almost it. So we have over here discontinuous replication, because we're making it in shorter pieces into the opposite direction, always in shorter pieces into the opposite direction. And we have also a name here, and there's not enough space. This is a piece where we have the RNA and the DNA over here. This whole thing has a name. It's called an Okazaki fragment. Mr. Okazaki fragment. That's an Okazaki fragment, right? And this part here is the RNA that has to be removed by DNA polymerase 1. And that's the new DNA, right? Okay, and the whole thing is called an Okazaki fragment. Yeah, and that is it. That's it. So to summarize, we have continuous replication down here. Discontinuous replication. Another word, lagging strand. It's lagging behind. The lagging strand. Okay, the lagging strand was discontinued. And so we have a lagging strand over here. The reason why we have it is because we have to stick to the 5' to 3' synthesis of new DNA. We can only do that by making it in the lagging strand in smaller pieces into the opposite direction. Okay, so that is it. So this was the replication fork. Okay, all the best.